For decades, research has focused on clearing amyloid-beta plaques, yet removing this upstream protein aggregate has consistently failed to reverse clinical cognitive decline. When examining post-mortem human cortical tissue, amyloid plaque density predicts only about 26% of a patient's actual dementia severity on clinical rating scales. The strongest indicator of cognitive impairment is the physical loss of excitatory synapses. As this trendline demonstrates, midfrontal and inferior parietal synapse density yields a near-perfect 0.96 correlation with the MADRS dementia rating scale. Historically, the scientific consensus treated the structural dismantling of neural circuits as a passive, late-stage symptom of generalized amyloid toxicity, rather than an active, targetable biological process. To restore memory, we must identify the specific, targetable mechanisms that actively dismantle the synapse. The research of Seth S. Margolis provides a biological framework that addresses this exact problem. His laboratory posits that Alzheimer's is the inappropriate reactivation of a normal neurodevelopmental pruning mechanism in the aging brain. In this model, the diseased adult brain is actively hijacked to dismantle its own structural connectivity and protein homeostasis. Margolis identifies two primary molecular executioners driving this collapse. The first is the Ephexin5 RhoA-mediated axis, which forces the physical retraction of dendritic spines. The second is the Neuronal Membrane Proteasome, or NMP, which governs protein degradation at the synapse. By defining these specific targets, this dual pathway model decouples synaptic failure and tauopathy from a strictly linear dependence on amyloid, mapping the actual machinery responsible for the physical symptoms of dementia. During early embryonic development, the brain uses a protein called Ephexin5 as a molecular brake. It actively restricts excitatory synapse formation to ensure neural connections only occur at the correct time and place. When a neuron is ready to form a mature synapse, this brake must be released. The FB2 receptor phosphorylates Ephexin5, tagging it for rapid degradation by the UBE3A ubiquitin ligase. Removing Ephexin5 allows the dendritic spine to expand and stabilize. In Alzheimer's disease, this precise developmental pathway is corrupted. Small, soluble amyloid-beta oligomers actively target and bind to the FB2 receptor on the postsynaptic membrane, triggering the receptor's destruction. Without FB2 to phosphorylate it, the degradation process completely halts. Ephexin5 pathologically accumulates inside the postsynaptic terminal. This buildup triggers the execution phase. Ephexin5 hyperactivates RhoA, a small GTPase that forces the actin cytoskeleton scaffolding of the dendritic spine to rapidly depolymerize and collapse inward. This mechanism is clearly visible in post-mortem human pathology. Across all advanced Braak stages of Alzheimer's disease, transcriptomic analyses reveal a 2 to 3-fold increase in Ephexin5 protein expression compared to healthy age-matched controls. To prove this accumulation actively drives the disease, researchers crossed transgenic Alzheimer's mice with Ephexin5 knockout models. The genetic deletion of Ephexin5 completely rescued dendritic spine density and cognitive function, even while the brains continued to produce massive amounts of amyloid. These results indicate that amyloid-beta requires Ephexin5 to execute its structural synaptotoxicity, identifying ARHGEF15 as a key pathway for the loss of neural connections. Ephexin5's role at the synapse is not static. Recent data reveals its behavior changes based on the neuron's immediate activity. In 2025, advanced live imaging of single synapses revealed that Ephexin5 undergoes dynamic, activity-dependent substrate switching. During plasticity-inducing events that mimic active learning, such as chemical long-term potentiation, Ephexin5 rapidly sheds its phosphate groups. This dephosphorylation event physically alters Ephexin5's binding affinity. It stops activating RhoA and instead shifts its target, preferentially activating a different GTPase called CDC42. Activating CDC42 achieves the exact opposite structural outcome. It promotes actin polymerization, growing and stabilizing the dendritic spine to successfully encode long-term memories. This creates a significant paradox. the protein responsible for collapsing synapses in the Alzheimer's brain, is physiologically required to stabilize those same synapses during normal learning and memory formation. While Ephexin5 dismantles the physical structure of the synapse, the second executioner of dementia triggers a localized collapse of protein homeostasis. This breakdown centers on the neuronal membrane proteasome, or NMP. Unlike standard proteasomes that float in the cytosol and require ATP, the NMP is an uncapped 20S complex physically embedded directly in the neuronal plasma membrane. When a neuron is firing, it rapidly translates new plasticity proteins at the synapse. The NMP acts as a rapid homeostatic modulator, immediately degrading a portion of this newly synthesized nascentome to prevent the neural circuit from becoming hyperexcitable and hypersynchronized. This degradation mechanism is heavily regulated by apolipoprotein E, specifically the E4 variant of ApoE, which is the single strongest genetic risk factor for late-onset Alzheimer's disease. This diagram illustrates the mechanistic failure. In a healthy scenario with the neutral ApoE3 variant, NMP complexes successfully embed in the membrane, capturing and degrading newly translated proteins like tau. But the ApoE4 variant physically prevents NMPs from localizing to the synaptic membrane, creating a severe localized proteostatic void. Without the neuronal membrane proteasome to manage the synaptic load, newly synthesized endogenous tau protein is left unchecked. It misfolds and spontaneously aggregates into high molecular weight sarcosyl-insoluble tangles. The ApoE4-driven depletion of NMPs lowers the threshold for this spontaneous tau aggregation by up to 50-fold compared to brains carrying the neutral ApoE3 isoform. By linking genetic risk directly to a failure in local protein clearance, the NMP framework provides an entirely amyloid-independent explanation for tauopathy. Neurofibrillary tangles arise directly from an ApoE4-driven localized proteostatic collapse. Understanding these downstream executors provides clear targets, but translating this molecular biology into viable clinical therapeutics faces formidable hurdles. The primary barrier is genetic pleiotropy, the reality that genes serve multiple, distinct functions across different tissues. The ARHGEF15 gene, which produces Ephexin5, is a prime example. Independent genome-wide association studies link mutations in ARHGEF15 directly to autosomal dominant cerebral small vessel disease, a major driver of vascular dementia. Ephexin5 is highly expressed in cerebral endothelial cells. Outside of the hippocampus, it is strictly required to maintain the F-actin cytoskeleton of the vasculature and regulate angiogenesis. Deploying a blunt, systemic, small molecule inhibitor to shut down Ephexin5 in the brain could inadvertently destroy the blood-brain barrier and induce severe vascular toxicity. To safely drug the Ephexin5 axis without triggering vascular collapse or erasing a patient's capacity to learn, we need highly specific pharmacological innovations. The solution lies in engineered neuron-specific PROTACs that degrade the target only in selected cell types, or biased allosteric modulators. These drugs must selectively block Ephexin5's interaction with RhoA while meticulously preserving its ability to activate CDC42. Translating the neuronal membrane proteasome model requires similarly precise pharmacology. Because the pathology stems from a loss of complex localization, traditional enzyme inhibitors are useless. Therapeutics must synthetically rescue 20S membrane trafficking directly to the synapse. By moving past the linear amyloid cascade and mapping the precise, bifurcated executioners of synaptic and proteostatic collapse, the Margolis laboratory has provided the sophisticated biological coordinates required to safely target and potentially save the aging mind.